about test cross and back cross simultaneously. First we discuss about test cross first, next we talk about back cross. So test cross means Mendel just tests something. That means he just tests a resultant to get some extra thing, that means some extra genes. So according to this concept, Mendel named it as test cross. So he don't know the F1 generation first. By taking the resultant, he just cross to get some parental gene. So we just talk about test cross first. So test cross means when the F1 hybrid, that means when the F1 hybrid is crossed with your homozygous recessive. So for getting the point, we just take an example first. So we just take as a parent, we just take two things. One is your pure tall plant and crossed with pure dwarf plant. So if we talk about its genotype, the genotype is for the pure tall is capital T, and pure dwarf is small t. We all know that. Nextly, as I told you that after the genotype, when we talk about the cross, we have to find out the gametes first. Nextly, the gametes. Here the gamete is your capital T, and here the gamete is your small t. Nextly, the cross takes place. After getting the gamete, the cross takes place, and the cross is capital T, small t. So it is the hybrid. We also call this as F1 generation. So this is the F1 generation, and these are the gametes and the genotype and the parent. So the definition told you that when F1 hybrid is crossed with homozygous recessive. So if we talk about this, the hybrid, it is the F1 generation, that means it is your F1 hybrid. So according to the point, F1 hybrid is crossed with homozygous recessive, not to the capital T. The reason is by taking your small t, it is the recessive character, and the point told you that homozygous recessive condition. That means it is your small t. So when we cross with the F1 hybrid to your homozygous recessive character or recessive parent, then it is called your test cross. So it is your test cross. In other words, we also told that when an unknown genotype dominant, why it is your unknown genotype dominant? Because when Mendel crossed these things, they don't know, that means Mendel don't know about this gene. He just get this gene, but he don't know that this gene is that means this parent is a pure dwarf, or it is a intermediate hybrid condition. He don't know the plant, about this plant. So for getting about this plant, he just tests something to get the result. And so this procedure is always known as your test cross. So he gets the unknown genotype because he don't know the genotype actually. He just tests it to get the genotype. Unknown genotype dominates to cross with its homozygous recessive. It is your homozygous recessive because it is the recessive character. We all know that it is your expressed character and it is your suppressed character. So it is small t is your recessive character of same character is called, because it is your same character, small t is called your test cross. So if you take this is your definition or this, it's your choice. So this is your test cross. Nextly, he just cross it. That means he just cross the resultant again. By taking two things, one is your capital T, capital small t is crossed with your another capital T, small t. That means by getting it, he just get this test cross and he just cross it. So here we just cross the capital T, small t with your small t, small t. And for this we just put a Punnett square first. So you just take capital T, small t. It is your capital T and it is your small t. And in other way, you just take small t, small t. There is not enough space, so we just write like that. So this is your, guess your male, this is your female. We just put it in a Punnett square. So if we plot capital T, small t, it is your capital T, small t. And next one is your capital T, small t. Here also. Here small t, small t. Here also the small t, small t. So if we talk about the phenotype, this is your tall plant. This is also your tall plant. That means for this gene, this is your dominant character and this is your recessive character. So this character is expressed and this character is suppressed. So the plant is a tall plant. So here also the tall plant. Here the plant is your dwarf because it is the pure dwarf plant and it is your dwarf. So Mendel, after getting the resultant, Mendel cleared about one thing that in monotypic cross, if we talk about the phenotypic ratio, Mendel taught that in phenotypic ratio, he get that two tall plants and two dwarf plants, and we also wrote it like that 1:1. So it is your phenotypic ratio. So in every monohybrid cross, in every monohybrid test cross, the resultant is your 1:1. So he just concluded that in monohybrid cross, the ratio is your 1:1. So in that way, in dihybrid cross, the resultant is your 1:1:1:1. The reason is in dihybrid cross, I told you that the ratio is your 9:3:3:1. That means there is 4 phenotypes. So here 2 phenotypes are there. So this is your 1:1. In dihybrid cross, 4 phenotypes are there. So it is your 4 types. That means 9:3:3:1 and 4 ones are there. 1:1:1:1. So this is your monohybrid cross and dihybrid cross ratio according to Mendel for the test cross. Nextly, he just think that I just cross the resultant character and get the genotype. That means I just cross the F1 generation with the parent and get the resultant. In that way, if we backly cross it, backly cross means when F1 hybrid is crossed with any one of the parent, that means he just crossed in both types. That means he just crossed the F1 hybrid with the capital T, and in other way he just crossed the small t. So he do one thing over here, that he just cross repeatedly. That means he backly cross it and also he backly cross over your small t. So the definition is when the F1 generation is crossed with any one of the parents. That means if you cross the F1 hybrid with any one of the parent, then the cross is known as your back cross. And the reason behind the back cross is the resultant. You just get the resultant of the same phenotype. So you just cross it, backly cross it. So there is another point arise from this condition, that every cross, that means all test cross, this is your test cross. When F1 hybrid is crossed with your homozygous recessive character, this is known as your test cross. So any test cross is a back cross. That means here it is also your back cross. That means your test cross is also a back cross, but your all back cross are not a test cross. So I wrote over here all test cross are back cross, but all back cross are not test cross, because it is your test cross. The test cross is your back cross, but it is a back cross, but it is not a test cross. That means every test cross is a back cross, but every back cross is not a test cross. He just concluded like this. Nextly, if we talk about every back cross, it may be your 1:1 ratio or may not be your 1:1 ratio. That means in every condition it is not bound to form your 1:1 ratio, but in test cross the monohybrid ratio is your 1:1 and dihybrid ratio is your 1:1:1:1. So he just gets this type of some result by getting your test cross and back cross. Hope this class is helpful to you. Thank you.